Hi everyone. This week's video is titled Pharmacological Tools Used to Promote Bone Formation. I found this article titled Drug Cocktail Holds Promise for Spinal Injury on Science Daily. Scientists at the Imperial College London revealed that two existing medications can boost the body's own repair by triggering the release of stem cells from bone marrow for spinal cord injury. According to the National Spinal Cord Injury Center, nearly 300,000 people are reported to have spinal cord injuries in the United States. The average age is about 43 years old, and 80% of victims to spinal cord injuries are male. The most leading cause of spinal cord injury is vehicular, closely followed by falls. The drugs utilized in the study are to be used after surgery or fractures. In the article, it emphasizes how after administration, bone marrow goes into a certain red alert that sends the stem cells into action. The two drugs in the article used are CXCR4 antagonists, a drug typically used in bone marrow transplants, and beta-3 adrenergic agonist, typically used for bladder control. The bone marrow of healthy rats are to release mesenchymal stem cells. Mesenchymal stem cells are multipotent stromal cells that can differentiate into a variety of cell types. These include osteoblasts, chondrocytes, myocytes, and adipocytes. Here, the researchers expect that they can promote the activity of more stem cells to boost the body's ability to mend itself. In the study, mice received a single dose 2 hours prior to blood collection. After AMD 3100 treatment, they saw an increase in the number of fibroblast colony forming units. The figure to the right shows in vivo near-infrared fluorescence images of the L4-L5 fusion site in the spinal cord at 3 weeks post-op. Below, we also see three-dimensional bone volumes. The beta-3 AMD 3100 treatment significantly increased bone volumes compared to both vehicle controls and the AMD 3100 alone. From these methods, researchers expect a single treatment with both drugs to increase healing, calcium binding to the site of injury, and bone marrow formation. They also saw a trigger of fat cells in bone marrow to release endocannabinoids that suggest a role in mobilizing stem cells and thereby promoting healing. The primary article of the study was very dense and a bit hard to get through because of the complex pharmacological dosages and drug names mentioned in the results and discussion portion. Even so, the researchers have quite a few goals for their future. Typical stem cell research studies that begin from scratch are very lengthy and expensive. This method is beneficial financially because the approach used in the study harnesses the power of the body's own stem cells. Also, the drugs used are already existing for other purposes. This confirms the safety of the treatment. All that's left is to start human trials, however, funding is needed to do so. Thanks for watching. These are my sources.